Yes, Professor Sarah, may you please unmute yourself?
Assalamualaikum.
Assalamu
Can you
alaikum.
hear me?
Walaikum salam. Yeah.
Can I start?
Yes, you are audible. You can share your video and your presentation also. I think this is time that we will start, and meanwhile the participants will be joining us.
Okay.
I don't want to be there
Okay.
for much of the time. Okay.
Okay.
I cannot keep you waiting there, so that we will start it. And with this, we'll start. Really, I am very much blessed and elevated to have you with us this morning. Very early morning, it's 9 over here, and people have just started their day after having sahar. We say that over here, the fasting sahar. Okay.
Right.
Yes. Now they are preparing for the day, and we have been joined by so many dignitaries. All of them are faculty members, professors, and some scholars also there in our participation. So most welcome, Professor Sarah. For my participants over here, I wish to just introduce a little about you. I don't have much of the profile with me about you. A little. That's your name. It's an interesting name. It's Professor Dr. Sita Sarah Binti Uthman, if I am correct in pronouncing it. So she is professor at Department of Cell and Molecular Biology, Faculty of Biotechnology and Biomolecular Sciences, University of Putra, Malaysia. She did her PhD from University of Glasgow, UK, and she has an expertise in bacterial pathogenesis, veterinary vaccine delivery, host pathogen interactions, cancer immunotherapy, infection, and immunity. So she is also co-chair of Science Leadership Working Group of Young Scientists Network, Academy of Sciences, Malaysia, and executive member of the Executive Committee of Malaysian Society for Molecular Biology and Biotechnology. Also a coordinator of the Microbial Biotechnology Laboratory. He is also associated member of Medical Biotechnology Research Group, Malaysia. This is a brief profile with me about Sarah, Professor Sarah over here. And I'm really thankful to her. She has spared her time, and she's this morning with us with her presentation with her presentation with us about in celebrating our this World Veterinary Day. I requested her because I somehow heard her her interest in veterinary vaccines, particularly HS. So I thought she'll be a better resource person for our this celebrations of World Veterinary Day. And that's why I have just invited her, and she has been kind enough to accept our invitation. So Professor Sarah, it's over to you. You can start your presentation and you can on your video also.
Alhamdulillah. So thank you very much, Prof. Zahur Pakpuri. That's one thing that I would like to apologize. I think I've updated Prof that my internet is a bit unstable sometimes, especially when I'm sharing slide. The university is currently upgrading the internet infrastructure. So will it be okay if I switch on my camera a bit later in case of disconnection problem?
As you wish.
Okay, right. Because I'm sharing a little bit of video, I'm scared that it's going to use a bit of a bandwidth on my part.
Okay.
Assalamualaikum and a very good morning to everyone. I would like first to thank Prof. Zahur Pakpuri for his kind invitation for me to share my insight today in conjunction with the Sharif Kashmir University of Agricultural Sciences and Technology of Jammu World Veterinary Day 2021 celebration. Like Prof said just now, just to clarify that I am a molecular biologist by training, but has been working alongside veterinarians throughout my research career. I am actually deeply humbled to be in this online platform together with fellow academicians, researchers, and also postgraduate students. Thank you for being here with me today as early as 9 a.m. It's 11:30 a.m. in Malaysia. It's heavily raining outside as well. Aligned with the theme of this year World Veterinary Day celebration, veterinarians' response to the COVID-19 crisis, I would like to share a little bit of what is going on over here related to our research in UPM. First and foremost, thank you so much, Prof, for the brief introduction. It's actually very lengthy already. I'm an associate professor. You can still call me Dr. Sarah, no check professor, at the Department of Cell and Molecular Biology, Faculty of Biotechnology and Biomolecular Sciences, University Putra, Malaysia. Maybe not many of us have heard about my university, the University Putra, Malaysia, or in short, UPM. If I may, I would like to bring you to a short tour to UPM by sharing a video.
Professor Sarah here again. I'm just we are unfortunate over here that we have been joined by our Dean Faculty, Professor Muhammad Ashraf Pal. May I request Professor Ashraf Pal for few words in favor of Sarah. Professor Muhammad Ashraf Pal.
Thank you, Dr. Zahoor. Assalamu alaikum wa rahmatullah. I, on behalf of the Faculty of Veterinary Science and Husbandry, welcome very heartily with the core of our heart, Associate Professor Sarah Othman from University of Putra, Malaysia, for having consented to deliver a lecture on the theme that witness response to COVID-19 crisis. It's a pleasure to have Professor Sarah on our online program. This is really a very good experience to have being shared with Dr. Sarah Othman. It's a really very good occasion for our students as well to be with Dr. Sarah, to have her experience shared with our faculty as well as our students. I wish all the best for the program. Please go ahead.
Thank you.
Thank you very much, Prof. So I'm going to bring you guys a short tour into the university. Please enjoy the video. Thank you very much.
Thank you, Sarah. It was nice to see your university. Looking very nice, beautiful campus.
I also really hope for future collaborations with Kwasiam as well, and I would encourage this webinar to be a bit informal, where you can actually ask me questions along my talk, if it's okay with Prof. Pampuri.
Sure, no issue at all.
Right, let me share my slide.
Yeah, please.
Can you see my slide? Right. So the University Putra of Malaysia is actually started as School of Agriculture in 1931 and upgraded into a College of Agriculture in 1947. So it started a long back. But however, in 1971, it has been granted as a university status and first called as University Pertanian Malaysia, Pertanian meaning agriculture in my language. And in 1997, it has been rebranded into University Putra Malaysia. Putra means the prince. So this year, 2021, marks our university 50 years celebration from 1971 to 2021. And for 2021, the university has been ranked as number 78 in the world for the Emerging Economies University Ranking 2021 by Times Higher Education. And for QS World University Ranking Top 50 Under 50, this year Malaysia has been, UPM has been ranked as number 10 worldwide and the first one in Malaysia. So I'm afraid for next year, we cannot join the top 50 under 50 anymore. So we have to fight in the real game. So just also in line with the, I think I've introduced myself a little bit now. I wanted to show that in our university, we have about 14 faculties and 1 institute that you can actually register for undergraduates and postgraduate programs. Our Faculty of Veterinary Medicine actually is the one of the oldest veterinary faculty in Malaysia. For now, we have 2, another one in the east of Malaysia. And I am from the Faculty of Biotechnology and Biomolecular Sciences. It's actually quite a new faculty. We are only about 11 years. So this is how my faculty looks like. Biotech at UPM. Perhaps one day you guys could actually come for a visit. So today's talks, like the theme of the World Veterinary Days 2021, of course, Jammu, under the COVID-19 pandemic, I wanted to share something very close to me, a disease that I've been working alongside veterinarians called hemorrhagic septicemia in bovine, how disease mitigations during the pandemic, and also the current and current pandemic vaccine development. So I'm sure all of us here knows Pasteurella maltosida. It's a very important veterinary pathogen, a gram-negative, non-motile, non-sporogenous coccobacillus or short rots. And it causes quite a number of diseases in every host that it goes into. 3 of the popular ones is avian fall cholera, bovine hemorrhagic septicemia, HS, and swine atrophic rhinitis. Pasteurella maltosida occur as a common cell in the nasopharynx of apparently healthy animals. For these particular studies, I'm going to focus on the bovine hemorrhagic septicemia caused by Pasteurella maltosida B2. So what happened with Pasteurella maltosida is that they reside in the submandibular regions of the cattle. And then when the cattle are happy, the bacteria are happy, they don't actually cause any disease. But things like monsoon season, a rainy season, for example, it stressed the animals or mobile or the transport of the animals or anything that caused the animal to stress, the bacteria actually produce endotoxin, then enable them to travel into the upper respiratory tract of the animal and cause, the first thing is a lung lesion that could actually kill the animal. And this is also called hemorrhagic septicemia. So the symptom that can be observed during this disease is the elevated temperature, loss of appetite, nasal discharge, salivation, labored breathing, with also swelling in the cheek, in the submandibular region. And the worst case, it's so acute that the death occurs within 17 and 24 hours. The thing is, this disease can be controlled if the animals are being vaccinated properly using the vaccine regime that we have. The problem in Malaysia is that the animals were being reared semi-husbandry condition or semi-wild, meaning that the animals are not contained in just one farm. They are normally reared in the village or in the rural area where you can find them along the roadsides or area that doesn't belong to the farmers. In here, it's okay. Also being observed in Southeast Asia as well. Countries like Indonesia, Thailand, Vietnam, and also Philippines also rear their cattle in this semi-wild breeding. What happened is, although the country have an annual vaccination programs, villages or the breeders don't really like to vaccinate their animal because of the so many disadvantages with the current vaccines, which I'll tell you after this. So annually during monsoon season, the villagers will lose their buffaloes to disease. And annually Malaysia at least lost 2.4 million ringgit by this death. Classic scenario is they sometimes vaccinated the animal in the farm, but they don't vaccinate the animals in the semi-wild area because it's difficult to call them back. And when that year the animal in the farm doesn't have any disease, they don't vaccinate it again the next year. So what happened is disease occurs in the semi-wild animal, it comes back to the farm overnight. In fact, the whole animals, the next 17 hours when the farmers wake up, all the animals have already succumbed to death. So the thing about HS is treatment require early detection of the disease, but the symptoms of the disease at first look like a common cold. So the only way to control the disease is via vaccinations. And in 1992, the country has started routine prophylactic vaccine via intramuscularly yearly using alum precipitated or oil-adjuvant killed whole cell vaccines. Both of these vaccines provides only short-term immunity. You have to inject the animal intramuscularly every year. So when the farmers doesn't want to do it, it does not effectively contain the disease. And it's unpopular with farmers because it's difficult. You need to get vets to go to the field to vaccinate every single buffaloes that they have, for example. And this vaccine is actually free. It's subsidized by the government. Even the veterinary service from the Department of Veterinary Services are also available for free. But it's just because, for example, like oil adjuvant, it has such a high viscosity when you actually injected intramuscularly to the cattle or buffalo, they began limping. And this actually, the farmers don't like them. It's difficult to sell the animal after that. And because of that, the vaccine have a low vaccination coverage. It's more on the understanding of the breeders as well. I wanted to bring you guys to a classic case in Malaysia. Like I said, this disease, although with the annually occurrence of its outbreak, still farmers are sometimes stubborn to vaccinate their animal. If any of you are familiar with Mowgli, Mowgli of the Jungle Book is a classic children's story books, and it has been absorbed into movies by Netflix and also a lot of cartoons in the televisions. So in short, Mowgli is a boy who loves animals, who actually speak to animals in these cartoons. So in Malaysia, we have our own Malaysian Mowgli called Muhammad Syukur Khamis. So Syukur, I call him. In this picture, he's a 15-year-old boy. He has been rearing buffaloes with his dad since he is 6 years old. He told the journalist that he loves animals and dreamed that one day to open his own zoo, and also he wanted to continue his dad's buffalo's husbandry business. Syukri has been popular in 2019 because photographers from all over the world has come to Malaysia to take his pictures in his buffalo's farm and enter worldwide competition, including the UNESCO nature photographies. And his pictures have won several of the competition as well. So what happened to Syukur? Did you think Syukur vaccinated his cattle? So this is what happened. In 2019, he won all these awards. This is one of the images that won the photography competition. So during the pandemic last year, although everyone is on quarantine, in August 2020, there was a first disease found in a village in Marang, Terengganu. In 9 September 2020, in Syukur's farm in Kampung Bagos Katong, Serada, Terengganu, which is at the east of Malaysia, first buffalo died. And at first, they thought it was a food poisoning. However, the sample was then taken by the Department of Veterinary Services and was confirmed that it is hemorrhagic septicemia. So the disease mitigation at this stage, the DVS will alert the 10-kilometer radius of breeders to quarantine the animals. At this stage, they have to call up all their semi-wild animals, stay in the farm and not go to a free grazing area. The DVS will come, the vets will go to the farms and look at the animal with symptoms and treat them with antibiotic, and also they vaccinate animals at this stage as well. Just few days after that quarantine, on the 15th September, in Shukur's farm alone, he lost more than 40 buffaloes from hemorrhagic septicemia. So he have about 110 before that, and 40 buffaloes just actually dead in a few days. So quarantines has been stricter, vaccination program has been executed, and on the 20th November 2020, when they do their final checking, the Department of Veterinary Services announced that the area, the 10-kilometer radius of Marang, Terengganu in the east of Malaysia are free from hemorrhagic septicemia. So how does this disease mitigation supposed to work? By right, each farmer should take an annual vaccination of double all adjuvant vaccines. This vaccine provide at least 1 year protection and a longer term immunity. However, like I said, it have a high viscosity, and because of that and the intramuscular route of application, it's difficult to administer, and coverage can only goes up to 70%. And during outbreaks, the DVS will actually treat the animals that show symptoms and antibiotics, and also supply them with another type of vaccine, which is alum precipitated vaccine. The advantage of this vaccine, it can be stored in room temperature, it have a very low viscosity, so easier to administer, and it have a fast reaction. However, it only have a very short-term protection against HS, and this lead to low-level coverage. So, like I said, annual loss of 2.5 million, it have a major effect towards our economy, especially in the milk and meat industries, just because of our poor bovine healthcare regime. This has led to loss of investors' interest to invest locally, and that led us to import at least 70% to date of milk and meat to meet the local demand. If we wanted to achieve the UNESCO SDG 2 for food security, we really need to increase our bovine healthcare so that we can have an investor interest to actually spend more money in these industries and actually stop us from, reduce our import percentage of meat and meat. With that in thought, since 2007, we have been working with this research. So research that I'm gonna tell you now is about vaccine development. This vaccine development, I will break them into 3 parts, right? The first part will be the development of live attenuated vaccine. The second part is the host interaction study. I wanted to see the molecular inside of the bacteria. How do they talk to the cells? Do they talk to the cell? And the part 3 is the bacteria-mediated delivery of DNA. So let's look at the first part of the research, which is the live attenuated vaccine. In 1999, they always say that the natural route of entry into the host mimics the pathogenic pathway. We need to develop a vaccine, a bacteria that is not killed, that is alive but attenuated or less virulent, so they can actually mimic the natural route of entry of the pathogen. This way, it can able to simulate a better immune responses. Together working with University of Glasgow, we have worked with a marker-free ARO-A derivative. This is a strain from Sri Lankan we call Pasteurella motocida B2 JRM12, where we have deleted the ARO-A gene that involved in production of aromatic ring in the shikimate pathway. And another strain we have developed locally called Pasteurella motocida B2 GDH7 that derived from a parent strain from Malaysia. And in this particular strain, we have mutated the glutamate dehydrogenase gene that involved in the conversion of glutamate. So we have GRMT12 and we have GDH7, one from Sri Lanka, one from Malaysia. So first, working together with the vets, we then help to test them in the field. So in the UK, we work together with Modern Research Institute in Edinburgh, and with Hobson et al. And Douglas et al., we look at how this live vaccine works. Can it protect the animal? So they sum up that via intramuscular, the cows are fully protected against challenge with the virulent strains via intramuscular. So they still use this live attenuated vaccine. They have tested several routes, but they proved that for this particular vaccine, it worked intramuscularly. At the same time, we've also tested the one in Malaysia. So this attenuated strain was recently received a Malaysia patent number, was recently patented. We have applied the patent since 2007 and finally it's granted. And this has been developed in 2007 during my master's study actually. And Rafida et al. In 2012, 2013 has tested in several buffaloes farm in Malaysia for a field study, and they found out that using GDA7 intranasally, they can produce or induce strong immunity towards in-contact buffaloes, right? So they vaccinated certain buffaloes and then leave the mingling. Also in-contact buffaloes were protected when they post-challenge with the wild type Plasmodium B2, even after stressing the animal with dexamethasone. So let's just sum up what Rafida did. So Rafida actually vaccinates several numbers, perhaps less than 5 of the animal, and then she mingles them with several of other animals. They challenge this animal, and then at least, let's say 5, they mix with 10, and then all 10 of these cattle and buffaloes are protected. What happened after that is these 10 commingling animals were commingles with another hundreds animals in those farms. And they then challenged all 100 animals, and surprisingly, they found all 100 animals were protected against this challenge. So at this stage, we know that GRT12 at certain extent provide a protection towards the animal via intramuscular applications, and the GDA7, the Malaysian vaccine strain, via intranasal sprays can actually protect mingle and co-mingling animals. And as a molecular biologist in that big groups, they were asking me, what else can we do with these strains? We already have a vaccine, we are patenting this, what else can we do? Because we know bovine disease is if you wanted to strengthen the bovine healthcare regime, there's other diseases that we have to address as well. Keeping that in mind, I was thinking that let's look at the molecular level of this particular vaccine strains. So that led to the second part of the study, which is the interaction study. I'm interested to see in vitro, meaning in the laboratory setting, if I take this bacteria and put them with the mammalian cells, the bovine mammalian cells, what does it do? Do they stick to the mammalian cells? Do they go inside? If they go inside, what do they do? So what we do is still working with these 2 strains, the Sri Lankan strain together with the one in Glasgow, the University of Glasgow, and locally we also work with the GDH7. So what we intend to do in this study is to look if this bacteria goes to the cell, does this adhere? And if it's adhere, does it goes inside? So that is where the adherent assays comes in. We mix the bacteria with cells. In this case, we choose embryonic bovine lung cells and also the bovine aortic endothelial cells. Both are bovine cells. And also we wanted to see if they stick to the cell surface, do they go inside by time? Can they go inside? What's the rate of them going inside? So that's the research question at that stage. So with the GRT12, we compare the GRT12 with the bovine Pasteurella moticida A3 and also Menhemia hemolytica A1, so bovine Menhemia hemolytica as well. And from this result, you can see that all of them adhere at a similar rate. So if you look at C, so this is just an image from one of the microbial pathogenesis paper. So you can see that somehow the Pasteurella induce phagocytosis towards the cells. And E is the Pasteurella moticida A3 and F is the Menhemia hemolytica A1. So they loosely adhere to the membrane of the cells. When you see invasion, when you want to see the internalization of the bacteria, you can see that you can find Pasteurella moticida resides in the vacuoles of the cell. So they get inside, but you cannot find the same result with moticida A3 and hemolytica A1. So you can see that the bacteria invade, but not the A3 and A1. We bring this work to compare with our Malaysian strain. So in this case, we have a Pasteurella moticida B2 wild type Malaysian strain, the GDA7, the vaccine strains of Malaysia, and also the Sri Lankan strain. So here we compare the efficiency of them to adhere, and all three adhere at a not significant rate, meaning they adhere more or less the same. However, when we look at internalization capacity, we can see that GDA7s are threefold better in invading inside these cells. In this case, we use a bovine aortic endothelial cells. We also look when the bacteria goes inside, how long does it stay in the cell? What happened to them? So we observe them when they go inside, they can actually persist in the cell for up to 7 hours with slow decrease, meaning they get lyse slowly. So I've become so excited at this stage, like, well, this is a vaccine strain and it can go inside and it stays. As a molecular biologist, I like to look at them like a car, bacteria like a car. This is a potential vaccine strain. They can carry other things as well. What if I ask them to carry a DNA with a different diseases of antigenic genes? Disease from FMD, for example, or tuberculosis, for example. So I then started to design in the lab together with my colleagues to part 3, which is a bacteria-mediated delivery of DNA. That means we also call them bacterfaction. A bacteria carry a DNA into the host cells. These works are still in vitro. So what we want to see here is, can we ask the bacteria to carry the DNA, goes into the cells, and actually release them? And when they are being released, what happened to the released DNA? Do the cells recognize them? So I'm not sure whether there's a molecular biologist in the room, but this is quite an interesting diagram. So this is what we call a dual expression vectors, and I named this as traffic light vector, PSRG. It have a 2 expression system. In a simple word, when this plasmid inside a bacteria will express a red fluorescent proteins, like a traffic light red. I'm in a bacteria, I'm red. The moment it goes into the mammalian cells, if the mammalian cell enable the uptake of this plasmid or the processing of the plasmid expressed from the eukaryotic expression cassettes here, you will see the mammalian cells to express green fluorescent protein. So why is it traffic light? Is when it's inside the bacteria, it's red. When it goes into the mammalian cells, the cells turns green. If the cells doesn't turn green, meaning the plasmids are being lysed, not able to be transmitted. If the cells express green color, meaning that the bacteria can shuttle the DNA from the outside environment into the mammalian cells and then make mammalian cells to express them. So we've published this work sometimes in 2013, and we work with JRMT12 first. So if you look at A here, this is all Pasteurella maltosida under confocal laser scanning microscopy. So that's why it looks a bit elongated because of the splicing. And this is a membrane-bound stain, the blue color, that overlay with the red fluorescent protein that the bacteria express. So now we know that all these bacteria carry the plasmid and express the red fluorescent protein. So if you look at my cartoon here, so the big one here is a cells with nucleus and lysosomes. So when we ask them to invade, they induce phagocytosis. Somehow, this is still unknown, and it goes inside and fuse into vacuoles, and somehow after time, they get lysed off. When they get lysed, because of the tiny size of plasmid, it escapes degradation and it goes travel into the cytoplasm. The mechanism of them traveling from the bacteria into the nucleus is still unknown, even on other pathogenic bacteria. So we view these samples under confocal laser scanning microscopy, and we look at this cell. So this is a cell stained with plasma membrane stain, a blue color. And then if you see the membrane ball, inside the membrane ball, red tiny dots here is your Pasteurella maltosida. At 3 hours, you can visualize that Pasteurella is inside, still inside. What happened at 3 hours when we look at other cells as well, some of the cells have Pasteurella inside, and when we switch the fluorescence wavelength to view green fluorescence, we can see the same cell that have bacteria here are already expressing green fluorescent protein. So this has proven to us that some of the bacteria has been released, and the DNA, the plasmid DNA, traveled to the nucleus of the mammalian cell and enable expression of the green fluorescent protein. This is actually good news at this stage. And then we view them again at 5 hours, and we can see these two phenomena. Some of the cells still having this bacteria, but expressing GFP, but some of the cells do not have the red fluorescent protein anymore, but continuously expressing the green fluorescent protein. So this has confirmed to us that this JRMT12 can actually deliver DNA inside, and also enable the mammalian cells, which in this case is embryonic bovine lung cell, to express the reporter protein. So we take this work back to Malaysia, and we wanted to see whether we can see the same thing with GDA7. So what we see here, the same effects. So you can see that here is the bright field cells that looks like under the inverted microscopes. And then you can see that intracellularly, these bacteria are inside these membrane-bound cells. And when you look them after 5 hours, you can see some of these cells already expressing GFP. So the same phenomena that we observe with the JRT12 strains. So we are so excited at this to know that the potential live attenuated vaccines can actually go inside and carry DNA around with an ability to express the reporter proteins. The possibility of us changing the reporter proteins, regardless who's expressing them, say the prokaryotic system or the eukaryotic system, can actually add on to target other bovine diseases as well. And then we are excited to continue this work. What happened is pandemic occurs. And the pandemic occurs in UPM, especially in Malaysia, quarantines happen. All labs are closed. University are completely closed. We have to stay at homes. We have to sacrifice some of our animals or just feed them. We cannot continue any animal works. Suppliers for labs cannot work as well. Students, even though they have registered for their masters or PhDs, they have to defer their semester. So the work has been stagnant. So we were thinking, let's go back to our strains. Let's characterize them. We've never actually looked through the profiling of our strains, the JRT12, the GA7, and also the wild type. So we bring our works in bioinformatics, in silico studies, and we look at the other potential antigenic genes that Pasteurella carries. So we look at the outer membrane protein profiling using a predictor in silico, and we also did some of the molecular profiling in genomic and proteomic. So these two papers you can access, and any papers that you would like for me to share, I would share in the future with Prof. So Prof can also share with everyone. So in here, we can see that each GDA7, the Malaysian vaccine strain, JRT12, the Sri Lankan vaccine strain, and also the wild type have completely different profile. You can identify them by using these two techniques. This is really interesting because we know they have a different deletion, different restriction of the genome that led to this information. So like I said just now, Alhamdulillah, we have filed for patent for this particular strain since 2007. And finally, I think somewhere earlier last year, managed to get it granted. So these strains are actually ready for commercialization. So in summary of my work, like I said, in a simple word, we, in our laboratory, together with University of Glasgow, we have actually obtained 2 live attenuated vaccine candidates, DGRT12 and DGJ7. Both are proven to be a potential vaccine strains, one basic with intramuscular and another one with intranasal applications. Both strains are cell invasive. They can actually adhere and invade the bovine arctic endothelial cells and also embryonic bovine lung cells. And both strains shows a significant efficiency of back-to-faction, meaning to carry DNA inside, release, and allows the eukaryotic expression system to work in the mammalian cells. So the current work here is very obvious. We wanted them to have not just one vaccine. We wanted a single spray to contain as much as possible vaccines so that it's easy to the farmers. They would like to use it. And if it's intranasal, it's easy. They don't have to call the vet. They could just get it in a bottle and then mix them and just spray their own buffaloes. And the fact that they don't need to spray every individual buffaloes, they can actually spray it in a number of them and commingling them to the semi-wild animals. This is the most important route that we wanted to achieve. So can you imagine if in the future we can develop a dual vaccine system with other diseases? We can incorporate foot mouth disease of bovine, also bovine tuberculosis. That is along the pipeline. So this work could not happen without these people. So I have the grants has been funded by the Ministry of Education Malaysia and also the University Putra Malaysia. I have worked quite a lot with the in silico studies with Kasasat University Thailand, University of Glasgow, Modern Research Institute, and some of the work that we did in the UK was with the Wellcome Trust. And all these 3 are all my supervisors since I did my master's. Prof. Zamri, Prof. Raha, I think if you work in bovine and also fish these days, you will come across a lot of Zamri Saad's name. Prof. Raha is a molecular biologist, Professor Zunitar is an immunologist. We have John and Roger here, both are retirees. It's all my supervisors in Glasgow. Chris is not here. Chris is also my supervisor when I did my postdoc in Morden. And also my collaborators in UPM, Dr. Jeff, Dr. Chia Svetlin, and my collaborators in Thailand, Dr. Tirasa. And of course, all my lovely students here. Without them, nothing of this possible. With that, I would like to share my thank you slide. Feel free to email me if you have research interest to work together, and also any information or any questions regarding my presentation. Thank you.
Thank you, Sarah, for your nice information. It was really a good information for us. For whole
I'm not too fast.
Yes, definitely. We have so many over the specialists in microbiology itself. We have a Professor Altaf over here. He's professor in the microbiology and virology. And in other words, all veterinarians, they do know somewhat about bacteriology, macrophages, and all that. So it's not alien to us. We are accustomed to all these technologies. But it was really nice to see that you have somehow modified this and you have gone for live attenuated instead of killed. We are still using this killed, rather alum precipitated vaccines over here also. Adjuvant vaccines we are yes using over here, even to 6 monthly also in some endemic areas. Right. I think the person which is really related to your work, that's Professor Altaf. I wish he will have some interaction with you. Professor Altaf, please, can you unmute yourself? Professor Altaf, can you please unmute yourself? Anybody otherwise? Professor Hamidullah Malik, sir, can you unmute yourself, please?
I think we got someone in the chat as well.
Yes. Assalamualaikum, ma'am, for the good lecture. That's the Professor Malik, Professor Azmat Alam. Can you please unmute yourself? Yes. Azmat Alam, sir.
Assalamu alaikum. Thank you, Professor Farah, for this very nice lecture. In particular, I am not actually, neither I am the microbiologist or immunologist. I am a poultry scientist. But what good thing I like, and what it has been going on in my mind also, that instead of going for repeated vaccination, first for one disease and then another disease and third disease, fourth disease, so every day we are going for vaccination for one disease or another disease. This is a good thing that if we have combined vaccines, like we have the combined vaccines for pets, we should have combined vaccines for our bovines also, so that all the disease, 3/4 disease get covered in one shot. And secondly, the delivery system, you talked about the nasal spray.
Yeah.
And this is a great thing, so that a farmer is at liberty, he can do it himself. He need not to search for a technical person who can administer the IM or some other subcutaneous route vaccines. So this nasal spray, every farmer can do it himself.
Yes.
That's a great thing, and that we need to get into all other vaccines, such delivery systems. Thank you so much for the nice lecture, and thank you, Dr. Pompuri, for roping in Professor Sarah for this very nice lecture.
Professor Azmat Alam, we are humbled over here that Professor Madan has joined this meeting. I am really elevated to see him in our. Professor Sarah, we have with us Professor Madan. He is internationally renowned scientist in reproductive physiology. So I will humbly request Professor Madan to unmute himself and just introduce here or some words of wisdom. Hey, I am humbled to see you, and really, I am really grateful to you, sir. Please unmute yourself. Yeah.
Oh. Hello, Sarah.
No problem.
Very nice to be listening to you and hearing
you.
from you about your exploits. It has been, I joined a little late, so I couldn't face with all what you said at the beginning. But it was very interesting to listen to your approach over into the animal diseases. As a matter of fact, this happens to be a week in which that we'll be talking about animal health. As a matter of fact, now One Health, with the Veterinary Day approaching on 24th. Day approaching on 24th. It was nice listening to you. Thank you very much.
Thank you.
My humble request to all the participants, can they just switch on the video so that I can have a click?
Prof, I think one question in the chat from Asif Sakit, is it? Yes. So you talk about combination of vaccines. Yes, if you use antigenic genes, for example, in the system that I've introduced earlier, you can actually put either prokaryotic system or eukaryotic system. Our futures, we actually also adopt these techniques with the cancer immunotherapy. So now we have the bacteria to also signal themselves so they know where to go when they go into the system. Let me share a little bit for our work with cancer. So let's say we are targeting colorectal cancer, you need to direct the bacteria to exactly goes to the cancer cells, cannot just any other cells. So what we do is we put a signaling system using the DNA so that when it goes inside the system, it's a targeted delivery. So I agree, other vaccines that you can combine, I suggested just now, you can actually get some antigenic genes from FMD, the foot mouth disease virus, and perhaps the BCG from bovine tuberculosis. That is among the pipeline. Hopefully, inshallah, the labs and the university operate 100% in the near futures with everyone's get vaccinated against COVID-19. And inshallah, we can try to achieve this together. I welcome collaboration. Please contact me for any student exchange or any collaboration. I think the university is quite an open arm with collaborations.
Yeah, I think Professor Azmat Alam is the person who can look into this about those MOUs and all that.
Okay.
Professor Alam can be
Sir,
have a right
sir,
to.
Pampuri sir.
Yes.
Pampuri sir. Yes.
Professor Pampuri,
Just,
nice to see you. Professor Pampuri, nice to see you and welcome you.
Sure,
And
sir.
I must convey my thanks to Azmat because he sent me a mail saying that there is going to be a webinar like this, and got witness to the presentations of Sarah, and that was very nice of Azmat as well as now very nice of you to have booked me in. A very pleasure to be associated.
Yes, sir. Wish you in our other webinars too there, please. I'm sending this on that physiology group, sir. I don't know, you are just taken it from there. Anyway, for rest of the days, we are still 2, 3, 4 days. I will send that to you personally, sir. Sure.
I also would like to inform on 24th. We are having a very mega seminar.
Yes, I got that flyer. I got that flyer. Your Haryana chapter, sir.
Yes, and I think that I would like all that students.
Sure, sir.
The students of your institution, maybe of other institutions, maybe Sarah's institutions also.
We have already circulated in our group, sir, in our faculty group. We have already circulated that.
You must have seen a galaxy of speakers.
Yeah. Yes.
And they're talking on various aspects and saying that what has to be done in the rural areas, especially that should be of help to us in understanding about this.
Probably for us, it will be very difficult to join that day because we are celebrating it a little bit physically. I mean, with all SOPs in place there, we are just celebrating that day in a physical mode. So probably will not be there.
You have a program in the afternoon?
Yeah, sure. We will join definitely in afternoon. Sure.
Okay.
Sure. Professor.
Professor Pampuri, I would like to just welcome in among the participants. We have Professor S. Pan. He's a senior professor and a well-known figure as far as LPM and poultry sciences is concerned. It gives me great pleasure to welcome him, and I thank him for sparing his time to be with us for this webinar.
I can't see him. Subhanshu.
Yes, sir. Yes, sir.
Professor Pan can unmute himself. Professor Pan, can you please unmute yourself, and you can just switch on your video also. Professor Pan. Anyway. So, Professor Azmat Alam, I think.
Okay.
Is it okay now?
Okay, we can. We can call it a day for time being.
We call it a day. I think it's our duty, moral duty, and to just pay our thanks to the speaker of the day.
Yeah, of course.
And you are the right person to be there on behalf of the faculty, on behalf of the university. Please, Azmat Alam, sir.
It gives me great pleasure to propose this vote of thanks on this second lecture of the webinar series, which we have organized as a part of World Veterinary Day celebrations. At the outset, let me thank our Dean, Professor Paul, for steering our this program, for being a leader, for leading us to this celebrations of World Veterinary Day. Then I would also like to thank Professor Madan, Professor Pan, Professor Hamidullah Malik, and all other great stalwarts of this veterinary profession who have spared their time and who are with us. I may not able to name all because I am actually presently I am driving. I have stopped my vehicle on somewhere on the road. So thank you all the dignitaries who are there in this webinar. Then and most importantly, thanks to Professor Sara from Malaysia, who has delivered a very nice and very short, crisp and nice lecture for us. I hope that many leads will be taken from this lecture, and I look forward for collaborations. I believe, Dr. Pampuri, you are searching for our student. International student exposure, that is the part of our NAHEP, that this can be one of the leads, and it can lead us to other destinations also, where we can at least send our students for this international exposure. And then we have international programs for the teachers also, and they can also find the collaborations there, and then they can also take advantage of this relationship that is being built here. I thank all the participants, all the students, all the scientists who are there with us. And lastly, I thank Professor Pampuri for arranging such a nice programs as a part of the World Veterinary Day. Thank you so much.
Oh, thank you, Azamat. I think as a host, it's my moral responsibility. Besides first that vote of thanks, I'm really thankful to all the participants, particularly Professor Madan, Professor Pan, and all our stalwarts who have already retired, but they have joined us this morning, despite being it Ramzan over here, and by 9 almost all are there asleep, but they have made this sure to attend this meeting in the very early morning, and then I cannot be there. The day cannot be there, I can say, complete without thanking the speaker of the day, that's Sara Othman. I'm really obliged and really indebted to Sara that she has spared a time and she has accepted our invitation and she has been with us this morning. Thank you, Sara. We look forward for your cooperation in seeing our students there and we have a program, but probably this time due to this pandemic, you are not accepting those students there because your institutes are also closed there. But hopefully, once it's over and international this travel is allowed, you may help us. You may cooperate with us in just getting our students exposed to your laboratories, to your setups there. That's the program with us, but we look forward for your cooperation there, your favor there. Thank you all.
Just wanted to check with Sarah. Is Professor Khalil Rahman around with you there in the university?
Abdul Rahman?
Yes.
Abdul
Was
Rahman
Professor
Omar.
of Reproduction and Genetics?
We have Abdul Rahman Omar. Is it the one?
No, this is, I'm telling a story of about 12 years now.
Could you maybe retired already? Is it in the vet faculty?
He was a vet and he was a very good and excellent reproduction man.
I think I've remembered him last time when I did my master's. If I've met him, I'll send him your regards.
Thank you. Thank you. Yeah.
Thank you all. Thank you one and all. So I am just ending the meeting over here. Thanks a lot.
Ramadan Kareem, everyone.
Thank you. Assalamu alaikum.
Salam.
Have a safe and good day.